Okay, so we're gonna talk about something called the dihybrid cross. Now you've all done, or at least most of you have done it in middle school. This big A, little a cross with a big A, and you've all done the idea that one, and we've discussed in meiosis why this is because you're going to give half your DNA to your child through a gamete, a sperm or an egg in a human. So that means one of these two, this parent will give, one of these two, that parent will give. So we represent that by having a big A, little a, having one of those two will be donated to the zygote, and then the other parent will donate one. So one of each goes in these squares. That's a zygote. That's a zygote. This is a zygote, and this is a zygote. A zygote, remember, is a developing fetus, and eventually it'll be turned into a fetus, and then eventually a baby will be born, if it is actually to be so. So this is the genotype of the full organism. This is a genotype of a full organism. This is the genotype of an egg. This is a genotype of another egg. This is of a sperm and another sperm. Okay. We're used to that. That is not a dihybrid cross. That's a monohybrid cross. But when we talk about dihybrid crosses, we're talking about two genes. You do have the big A, correct? And you have a little a, but maybe you have a big B and a little b as well. And perhaps you marry someone. That's what the cross means, to you mate, right? Mate, to cross. Can be maybe that has a big A, but maybe has little b as well. This could be, each of these could have their own three options. You can have the heterozygote, which is one we covered in yesterday's question, will be one of each, right? A dominant and a recessive. Or you could have the homozygote that could be dominant, or you could have the homozygote that's recessive. Either way, there's three options for each gene. If you have two alleles, one being dominant, the other recessive, then there's three options for an individual to have on each gene. Each different letter is a different gene. So A controls height, let's say, and B controls color, or any number of traits. There's tens of thousands of traits that are controlled by genes. So it's good that we know that all these progeny are going to be tall, but how many are going to be tall in green? How many are going to be tall in yellow? How many are going to be short in green? So that's two traits, right? Two traits. And they are independent of each other, correct? It's Mendel's laws say that one gene doesn't depend on the other. By the way, we know that that's not true anymore. We know that there are a lot of genes out there that do depend on each other. But in general, it's still true. We can go with it. We can go with the idea and think about how things are inherited and how. I have no words for what's going on here. It's like watching a clown car unload. It's so, yeah.
So if it were to be proven not true as found by recent scientific discovery, would it not be considered a law anymore?
When I say not true, I mean not 100% true. So like laws, as we said, are mostly true. There's very little that a human mind can grasp that can say this always happens. The word always happens, tough, very tough. This is how it usually happens, is what we really should say. This is what we've experienced, how it usually happens. But there are a number of exceptions. We've already talked about a couple of them, codominance and incomplete dominance. That breaks Newton's law, not Newton, I keep thinking because we talked about it, but Mendel's first law of genetics, which is that each parent has two alleles, one is dominant over the other, and we'll give one of those alleles randomly to their offspring. We know that's not 100% true already because we have incomplete dominant and codominance. So we call that kind of inheritance as non-Mendelian. So Mendelian genetics is just dominant recessive. Non-Mendelian genetics is really a better description of what's going on. Very similar to the concepts that I discussed with you previously, which is Newton's laws of motion. We still call them laws. They're still accurate in most conditions. But really, we know Einstein's laws are more accurate. So really, the more complex understanding requires development. So we start with Mendel's laws, and then we move forward. Okay? So we're going to cross these two, and we want to know what is the combination that we're going to get. Well, the way we do it, and you know what? Let me make both of these, if you don't mind. I'm going to make both of these heterozygous for both genes. For both the A gene and the B gene, they're heterozygous. This could be the father, and this the mother. All right? Those are just generalized symbols for male and female. All right. So how do we know how many different gametes can this father make? Go ahead and try to figure it out. Do it now on your own. How many different gametes? I'll give you a couple minutes. See if you can do it quickly. Any math majors here? Any math interested in math? Love math. So you can do this quick, can't you? Not 16. You can't do it quick? You thought you love math. Don't you love math? Okay, well, yeah.
Is it 9? No.
We'll find out. I didn't say yes or no on that. Well, how'd you get 9?
I just did looked at all the types.
You start kind of counting them up in your head?
Yeah.
Okay, yeah. 8 or 9, yeah. Have you ever heard of something called FOIL? Yeah, you can use FOIL here, yeah? First, so we go ahead, just like in algebra, first, so you can make big A, big B. That's one kind, right? That's one gamete. You can take and make a big A, little b. That's another gamete, correct? All right. Then you can make little a, big B. And then finally, you can make little a, little b, correct? So you can make 4 gametes. It's not always true. It can be less, but that's the maximum you're going to be able to make, and because they're both heterozygote, right? So some max differences. Oh, let me plug this in real quick. So your question is a good one. How is it that you have 2 alleles in each gamete? Aren't we supposed to have only half? So shouldn't there only be 1 allele in each gamete? The answer is yes and no. The answer is there should be 1 allele of each gene in each gamete. So there should be 1A and 1B, and that's exactly what you have. Remember, there's 46 chromosomes. Instead of, meaning 2 copies of each chromosome. So at the end, you're going to end up with 23 chromosomes in each cell, each gamete, but that means thousands of genes, right? But only 1 copy of each gene, not 2. Are we good? All right, so this is excellent. It's good thinking. So if we did that, if we did this FOIL for the mom, we'd get the same thing, wouldn't we? Correct? Because mom has the same genotype, so there's the same possible babies, or I'm sorry, eggs that as the sperm that the father can make. So it's same possible combinations. These would be sperm. Now they're gonna make eggs. This male is gonna mate with a female. It's a human, so it's gonna possibly make it little a, big B. That's an egg. Big A, big B. That's a possible egg. Again, possible egg. We don't know which of these possibilities is actually going to happen. We can make the mother could give this child a big A, big B, or the mother can give a, little b. I don't know why this. I think this cord is. I see. All right. So these are the possible zygotes that the parent can make. All right, so if this sperm and this egg came together, if this sperm and this egg each delivering 1/2 of their DNA. Let's go back and look at mom and dad again. Mom had one big A and one little a, and dad had one big A, one little a, one big B, little b each. Now you're giving one of each of these randomly, so we have to figure out what is the random possible distribution, which is why you do a Punnett square. So here we're doing little a, big B, big B. Notice I kept the letters the same, together. The A's together and the B's together, correct? I like to put the capital letter before the lowercase letter because that's how I read. All right, usually that's what we do. All right, just so you know. So big A, big B, big B. So you guys go ahead and do it while I'm doing it. I think if you see I do something different and you're confused, ask me. But try to do it on your own right now. All right, next is yeah. We will in a minute, but wait till we do it together. Sorry, it's hard to write this with your finger. Are you all done waiting for me? Dang. How does that look? Yours look something like that? Okay, so for the sake of clarity, I'm going to go ahead and say, let's say big A equals red. Is a red color. And let's say little a equals a white color, just because we talked about it yesterday, and let's talk about it that way now. Let's say we know that big B is tall, and little b equals short. We're keeping it simple. These obviously can be any trait. It could be sickle cell. It could be a lot of different traits. It doesn't have to be something as simple as color and height. In a plant, because in humans, height is not controlled by dominant or recessive, except for dwarfism, maybe giantism, but I'm not sure about that. Those could be dominant recessive, but all the other genes involved it's a multi-gene inheritance. So how tall you are is not tall or short. It's all kinds of heights. Yeah.
So is this representing a multi-gene inheritance?
No. This is two genes. How are these two going together? These two are moving together. These two are this is how they randomly assort. So how many tall and green plants are you going to get? So each one is a multi-gene inheritance, means there's multi-genes like you see here, but it's affecting only one trait. In the case tall, in the example we just used. Here you do have two genes, you're right, but they're each impacting a different trait. One's impacting color, the other height. All right, so let's go ahead and take a look at this. Let's do color first, either red or white. Tell me, is this one red or white? White. It's white. I'm going to go down. The next one, is it red or white? Red. The next one? Red. Next one? White. Next row? Red. All right. We're all good with that? How we know that? How do they know it's red? If you're confused how you know it's red, you need to let me know. All right. Then the rest are white, correct? This is only two options. Oh, I already said white. Why did I do that? I don't know. All right. Now let's write tall or short. Let's see. Tell me if it's tall. First one's tall. Hopefully the pencil works so I can, yeah. First one's tall. 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 So now we can easily do our phenotypic ratios. All right. How many, I don't know why I put 2 white dots here. All right. How many tall white flowers, plants do we have? Tall white, tall white, tall white. So it's 3. How many how many short white do we have? 1. So 3 to 1. How many tall red do we have? 9. How many, let me write that down. It's the first 3 are tall, short, and white. Then 1 was white and short. This is short and white. And there's 9 what? Red and tall. And the next is what? Tall and white. No, we did tall and white. Short and red. Is that right? How many short and red are there? 3. Now the easiest way for me to say is that, let's say it's instead of 3 to 1 to 9 to 3, let's say 9 tall red to 3 heterozygotes. So tall, let's just say 3 tall white to 3 short white to 1 short white. Oh, wait. It should have been short red. Short white. Now, why did I organize it this way? Because what I did here is the first one where you have both traits are dominant. And just randomly, you see more of them. You'd expect more of them randomly. Does that make sense? If with this one, you have one dominant trait and one recessive trait. And then the next one also is one dominant trait, one recessive trait. Again, you'd expect that because as you get it, you're going to see it more often because it has, if you get it, you see it. Make sense? And then you only have one, the least frequent one is going to be the double recessive. Is going to have the least frequent. So you get 9 to 3 to 3 to 1. That's the ratio you'd expect. Where the 2 double dominant is going to be the most prevalent. The double recessive is going to be the least prevalent. And then the 1 dominant, 1 recessive is going to be in the middle. As far as frequency, just randomly, just by numbers. Now, why do I say just by numbers? What if the tall flowers, as soon as they got tall, they got eaten. And if they're red, they attract the herbivores even more. So the tall red flowers get eaten first. How many How many tall red flowers are you really going to see in a population?
It's gonna be less.
It's going to be less. That's called evolution. It's called natural selection. So what's going to happen is that this is what you'd expect to see if it was just random. This is just random. If nothing else was impacting the population, these are the percentages you'd expect to see. However, we know that in the real world, that's not what happens. Yeah,
Is that why dwarfism, even though it's dominant,
you got it.
natural?
You got
makes
it.
sense.
Even though dwarfism is dominant, we don't see a lot of dwarves. I mean, I've met a couple dwarves in my life. I don't know a lot of small people. The reason is natural selection has held back their dominant gene. In modern culture, it would be sexual selection. In the past, it would be sexual selection, cultural selection. As you know, in the Sparta, if you were born with a malformation, they threw the babies to the wolves, literally, alive. Which really, I'm like, at least cut its throat. Why would you? Like, just let the wolves tear it apart? That seems cruel to me. But, yeah.
So, in the case where you said tall white flowers are that heated, or tall red flowers are that heated, would the short white flowers then be more popular?
Yeah, and so you'd end up with what? What would the impact would be? Only the short white ones would be. So you'd see a lot of short white ones, and over time, would there ever even be any red tall flowers? Probably not. You'd probably end up with a lot of short, and a lot of white, a lot of short white, mostly short and white in the population, and those would be the genes. Would also be mostly short and white. Because of that. That's what we call evolution. The frequency of the genes changing is called evolution. All right. So, all right. And that's a dihybrid cross. Now, there is a genotypic ratio. Somebody asked, should we do the genotypic ratio? Let's just for fun, let's try to do a genotypic ratio of this chart. I doubt anyone will ever ask you for it but it's easy to do. You just got to read. So how many total do we have? 1, 2, 3, 4. So 16 total. So we got one little a, big B, big B. Is there any other little a, big B, big B? No, there is not. Not that I see. Anybody see one? I could be wrong, but that's up. All right, so that's little a, big B, big B. What about big A, little a, big B, big B? Big A, little a, big B, big B. There's one here. There's another one here. Any others? Are we sure we're good? So two. There's three. Where's the third one?
Under the first one you crossed out.
This one here?
Yeah.
And this one? No, this is big A, little a, big B, big B.
No, the third one, in the first row.
No, it's
That's too little.
it's a big and little. So two bigs and one heterozygote for A and homozygous dominant for B. There's only two. All right. Now, what about big A, big B, big B? What about big A, big B, little b? Two heterozygous for both alleles, both genes. Big A, big B, little b. I got one here. I got one here. 1, 2. Where's the third one?
The third one, row.
Fourth row, second.
This? Oh, this one. We're all good with that? 1, 2, 3.
Four.
The fourth column, second row.
Wait, this one? Oh, fourth column.
Yeah, right there.
Second row. All right, so 4. We're all good with that? So we got 1, 2, 3, 4. That's big A, big B, little b. Now little a, big B, little b. 1, 2. And what about big A, big B, little b?
Oh, 1. 2.
Where's the second?
It's in the third
Oh, I see.
column,
Yeah, I see
second
him.
row.
I see. So this one, and this one. Big A, big B, little b, and there's 2. And big A, little b. So one of those. And big A, little b. And then little a, little b. So there's a lot of different ways to get these 9 to 3 to 3 to 1 phenotypes, right? There's a lot of different genotypes involved. It's a 1 to 2 to 2 to 4 to 2 to 2 to 1 to 2 to 1. Is there anyone, you think anyone would ask you to memorize that? No. I mean, it's kind of like your locker combination, but I suppose. But 9 to 3 to 3 to 1 is the one you really should be very familiar with. If you're going to commit anything to memory, just try to remember this. And try to remember that 9 is a double homozygous dominant and that 1 is a double homozygous recessive and the 2 3s are heterozygous for at least, what am I saying? Not heterozygous, homozygous. Nothing to do with that. 9 is the double dominant. The 1 is a double recessive. And the 2 3s are dominant for one, recessive for the other. All right. So that's a dihybrid cross for unlinked genes. When you're done with your interactive activity, we'll talk about linked genes.
How long is interactive activity?
Shouldn't be more than 5 or 10 minutes, I would expect.
It doesn't.
Uh-huh. All right. So when we talk about phenotypic ratios and genotypic ratios, it's not just for figuring out the ratios. We can predict and we can calculate probabilities. You can all stop talking now and pay attention so we get this. So there's 2 rules of probability. If A and B are unconnected, and what that means is that one doesn't influence the other, right? These 2 events are separate. All right, so it's not, some events depend on each other. If these two are independent, this is for any probability. Like for instance, bingo balls. Like bingo balls, like lottery balls. What's the probability of getting 3, 18, 29, 43, 29, or 28, 4, 10 in a row? You can calculate the probabilities for that. So you follow this rule. Separate events is the probability of A or B. Either probability is equal to the probability of A plus the probability of B. So this is the probability of getting A or B. So if you're going to get A or B, Then you add up the probabilities of A and B together. So if you have what are the odds? What are the odds of getting ahead on a quarter? Half, 50-50. So what's the probability of getting A or B? No. 100. Probability of getting A or B is 100% because you only got two. Right? Does that make sense?
No, it doesn't.
It's like both of them, guys.
So what's the probability of getting... All right, so try listening. So let's try again. I'll draw it out. You're still talking. It's like, it's so exhausting. You have a head and you have a tail. What's the probability of getting ahead? 50%. What's the probability of getting a tail? So what's the probability of getting A or B, a head or a tail if I flip it?
Then that would be 100.
100%, because the word is or. How did I get that? I added up. You're still talking. Haven't stopped. 50% the probability of getting A plus the 50% is the probability of getting B, which equals 1. This is me changing percentages to decimals. Okay? So that's what's called the sum rule. The sum rule. Because you're adding. That's when you have or involved. It's important. It's going to be in word. These are going to be word problems. So you got to know your language. The word or means I'm adding the probabilities of the options. Then you have the product rule. What's the probability of getting A and B? No, well, yes, that's true. But that's not what they mean. So, oh, did I say I put plus? I don't know why I put plus. So just copy what was above there, I guess. So, what that is when in this case, and they don't mean getting them both at the same time. They mean if I'm flipping the coin, all right, if I flip the coin and then I flip it, I get heads, what's the probability that I'll get heads again?
25%.
No, 50, it's 50% to get heads. Every time I flip it, it's 50%. Every time I flip it, it's a 50% chance. But what are the probability that I'll get heads and heads? Two heads in a row.
1 in 4, yeah.
No. I don't want you to think of it that way. You're right. It's not that you're wrong. How would you calculate? Okay, let me ask it this way to make it. All right, Mr. Smart Aleck. What's the probability of getting heads 15 times in a row?
50% times.
So you do 50%, 0.5 times 0.5 times 0.5 times 0.5. How many times did I just say? 10. I'll just say 10. 10 times in a row. 3, 4, 5, 6, 6. Shh, because people don't understand power. 7. Stop. Not everybody understands everything. I'm trying to take it one step at a time. Jeez, just go with what I'm saying. 1, 2, 3, 4, 5, 6, 7. Yes, there's all kinds of shortcuts. 8, 9, 10. So do the math. Somebody pull out your calculator and do that math.
0.098. 0.098.
I don't think that's right.
If you do the percentage,
Do 0.5 times 0.5 times 0.5 all the way across.
and it's 0.0098.
0.0098 is the number. Now, again, there are shortcuts, but I'd rather not do shortcuts because not everybody follows along. So if we multiplied the probability of getting this many heads in a row is that number. So it's 98 out of 10, 100, 1,000, 10,000. 98 out of 10,000. You'll get 10 heads in a row 98 times out of 10,000 attempts. So it's not very likely. So if you want percentages, how many out of 100, then you would multiply this times 100%. And what the odds of you getting 10 heads in a row is 0.98%, less than 1%. It's less than 1% chance of getting 10 heads in a row. And you can multiply that, you can do that forever with a calculator, and it's easy, right? It's called the product rule. This is when you're trying to find out, are you going to get head and head and head and tail and head. So every time you add up when you get the percentages of each individual event, you multiply them all together. If you want to know what are the odds of me getting this or this, two different events, one here and then another one here, or that happening here, then you add them. Any questions? Yeah.
So what would be the odds like you get head one time, tails another, and then tails again?
You want that specific order?
Like the formula.
Heads, then tails, then heads? The odds of one event, heads, times the odds of getting tail, times the odds of getting head again. Guess what? The same thing. It's the odds of each individual event multiplied times each other. If it's the word you're using is and.